Welcome, welcome. It's our final lab. It's about time. Yay! Right? So this is the last lab, polar bonding. So remember, polar covalent means unequal share, of electrons, and if it's the right structure, you can get a polar molecule. So water is polar, for instance, and like dissolves like, polar things mix with polar, non-polar things mix with non-polar. So that'll come important. We'll test it with the dyes. First thing I do is that we're going to make making slime. Actually, this is all stuff that's in the lecture course. So now we're going to make the slime. Polar bonding, slime time. Yes. So half a gram of the gum, and then 50 milliliters of salt water. So to dissolve this stuff, and then mix it up till you get the gum to dissolve. Then you put 4 milliliters of the borax in there. And then you stir it and it becomes slime. So actually, it's kind of fun. You get the stuff and you get the slime, and you can kind of bend it, and you can like rub it in the balls. You can squeeze it, that kind of stuff. It's fun to play with. So have fun playing with it, that kind of thing. So and then when you're done, wash your hands. Yep. Don't eat the slime. But now what you do is test some of the inks, the water-soluble inks. These are highlighters and pens and permanent markers, newsprint, dry erase markers. These things are not soluble in water, so water-soluble ink. Do that, and then take some newspaper. So grab some newspaper. It doesn't have to be like the Honolulu Advertiser. It can be anything. It can be the Honolulu Weekly or something. Whatever newspaper you want, just grab, and then just try the slime. Try the silly putty, see what it works, and then this will tell you rules of the solubility, what's being attracted to it, and various things. So that's more of a fun thing right there. So let's talk about ink chromatography. So you take your filter paper, so you have the round thing of filter paper, and then you put some ink spots on it, and then you cut a section in the middle, so half piece of filter paper, and fold it in half several times so it makes a narrow wick. So take it so it's nice and like a long sort of sheet of paper. Put it in a hole, and then stick it down into the hole in the filter paper, and then place that on top of the beaker as it's shown. So the water should wick. It should draw up and go to the ink spots, and then if the ink is water soluble, it'll separate out, and that's called chromatography. Why do we care? What do I care about chromatography? That's how you get genes to separate for DNA. Like if you want to do a DNA assay on somebody, you do it like this. You do it like chromatography, for instance. So do that, and then record your data. So here's slime observations. You had fun, right? So see what worked, what did not work. For these hypothesis. So silly putty, is it water soluble or water or not? Should let you know about the silly putty. And so chromatography observations, tell us what you saw. And I want you to take a picture of this after. These are fun. It looks kind of cool. A lot of times the inks, you take like blue ink and like, whoa, what color is that? It'll kind of separate in different colors. I want you to take a picture of this after the water gets going up there. It might take a little bit of time, but send me that picture. That's kind of cool. I always like that. So send me this picture, see what it looks like, and then go through with the chromatography. And so with this, so the idea from the silly putty tells you an idea which dyes are water soluble and which ones are insoluble. The water soluble dyes are going to ones that are separate out. So that's the thing. You're going to hypothesize, and your hypothesis could be wrong. That's okay. You don't have to go back and change your hypothesis. It's not going to change your grade at all. It's more that I'm more interested in what goes on inside your head rather than what goes on this piece of paper, right? So it's more important that you use the scientific method properly. So I'm not going to check to make sure, you know, everything comes out the way it should. So this is science. It almost never comes out the way it should. So this is the end of the lab, you know, and as always, you know, be safe, be very mindful of safety for each of these labs, and hope you enjoyed this semester if you're doing these in sequence, and I will see you later. Goodbye.